Hello, and welcome to the Drug Discovery World podcast, a podcast covering topics around drug discovery and development, pharma and biotech. My name is Giles, and I'm here to take you through this episode. Today's episode is taken from our winter 2018-19 issue, and is titled Systems Pharmacology, When Multi-Targeting is Advantageous. And this is part 2 of 2 episodes. The article was written by Dr. Kirkwood A. Pritchard Jr., Dr. Dustin P. Martin, and Dr. Stephen Naylor. So now on to the main article. Systems Pharmacology, When Multi-Targeting is Advantageous. Systems Pharmacology. The old model of one drug, one target was developed on the assumption that specific drug treatments would be superior due to the absence of off-target side effects. However, the promiscuity of drugs that bind to numerous targets is now a well-characterized phenomenon. Recently, it was estimated that promiscuity rates varied from 6 to 28 individual targets per therapeutic drug. It is noteworthy that, armed with hindsight, most blockbuster drugs, one drug, one target, are pleiotropic in nature. In other words, the drug has both numerous beneficial and adverse effects on the patient. A well-known example of such a drug is aspirin. The drug manifests effects in the treatment of inflammation, pain, fever, assorted cancers, stroke, and cardiovascular disease, but is also associated with gastritis and bleeding. Aspirin was recently reported to have an estimated 23 different putative targets, possibly explaining the pleiotropic properties of the drug. A 21st century example of another widely used pleiotropic drug is the statin family members. In both cases, the one drug, one target model was used originally in the development of such therapies, without the insights of drug promiscuity. Definitions. The growing body of evidence from drug promiscuity and systems biology studies indicated that the magic bullet, one drug, one target model was simplistic. In part, this is due to the compensatory mechanisms and redundant functions present in biological systems, facilitating a resilience to single-target drug perturbations. Currently, it is understood that many drugs bind to multiple targets that in turn participate in and are associated with multiple biological processes. This property is referred to as drug polypharmacology. The term was applied originally to off-target adverse effects of the drug. More recently, the definition has morphed to reflect the exploitation of these characteristics in a more beneficial manner. Polypharmacology is now defined as the design or use of a drug to either 1. Simultaneously interact with multiple functionally related targets that act together, or 2. Inhibit targets that differ functionally from the primary target of the drug to produce additional advantageous effects for the individual patient. Our understanding of human complexity delineated by system biology studies, combined with recognition of drug promiscuity and polypharmacology, led to the advent of systems pharmacology. This latter approach requires a comprehensive molecular and cellular profile of the healthy individual or population, systems biological profile, the diseased individual and population, systems pathobiological profile, and the corresponding response of the individual or population to a drug, systems pharmacological profile. The resultant comparative datasets from systems biology versus systems pathobiology analyses and the corresponding drug effects profile constitutes a systems pharmacology approach. In addition, the differences between these various states of the patient or population are referred to as systems response profiles. The fundamental principles for the one drug, one target model are essentially the same as a systems pharmacology approach. However, at the systems level, a multitude of targets, biomarkers, and diagnostic components are all considered. The resultant effect is a more comprehensively defined health disease adverse framework of the individual or population, as well as a safer, more efficacious drug. Systems pharmacology is predicated on the polypharmacology of therapeutic drugs. So what is the difference between the two terms? They are both used as descriptors to describe efforts to overcome the one drug, one target model. Polypharmacology refers to a one drug, multi-target concept as utilized in drug repurposing. However, systems pharmacology refers to a broader concept best captured as one drug, multi-targets associated with pathways and networks. Thus, systems pharmacology is based on the rational design of drug therapies using information based on molecular, cellular, and physiological complexity. This type of approach produces systems pharmacology drugs rooted in molecular interactions between a single drug and multi-targets present in defined pathways or networks. Combination therapy versus systems pharmacology drugs. We've discussed the limitations of the one drug, one target compound that includes safety or toxicity and side effect issues, as well as limited efficacy. In the case of multi-drug, multi-targets, i.e. combination therapy, such an approach is designed to elicit synergistic effects brought about by different mechanisms of action by more than a single drug. In this context, systems pharmacology is a one-drug multi-target model, combined with a pathway or network analysis that should significantly enhance efficacy and reduce safety and toxicity effects. Anigoro et al. Have suggested that while there is highly significant therapeutic relevance of combination therapies, there are potential advantages of targeted therapy based on a single drug that modulates the activity of multiple targets over single targeted or combination therapy. Some of the advantages of systems pharmacology drugs over combination therapies include: 1. A single systems pharmacology drug with multi-target activity may have a more predictable, therefore superior, pharmacokinetic profile compared to a number of individual drugs administered in combination. 2. Acute toxicity may be enhanced in more non-selective combination therapies. 3. Adverse synergistic effects may be more pronounced in combination therapies. 4. The probability of developing target-based resistance to multi-target drugs is statistically lower than is the probability of developing resistance against single-target drugs. 5. The administration of a single systems pharmacology drug results in a more consistent and predictable ADME profile. 6. Drug-drug interactions do not exist in a systems pharmacology regime. And 7. A single agent binding to multiple targets might be easier to develop given the regulatory requirements showing safety and efficacy of a drug combination. For all these reasons, the development of single drugs with a desired multi-target profile defined by pathway or network analysis do offer a compelling, cost-effective alternative to drug combinations. Historically, drug combination therapy has been more extensively explored in the clinic than the use of single multi-target drugs. However, both approaches are likely to yield single drugs or drug combinations with improved safety and efficacy profiles. In either case, key to success will likely be the informed selection of suitable biological targets and molecular pathways or networks that need to be modulated with drug molecules. Implementation of systems pharmacology. Levy has discussed systems pharmacology drugs in the context of the substrate enzyme lock and key model. He has suggested that a multi-target drug could be conceived as a skeleton or master key capable of simultaneously unlocking several locks. However, he subsequently gave the opinion that such a model was simplistic and did not adequately describe the effect of a drug on multi-targets over a prolonged period of time due to significant up and down regulation of gene signatures. A detailed comparative systems analysis, i.e. the systems response profile consisting of healthy versus pathobiological versus drug effect systems biology analyses, is required to produce systems pharmacologic drugs. But how to efficiently go about both rationale target selection and drug candidate design in such a complex system? Recently, Ramsey and colleagues discussed these issues and made the following salient points. 1. Target selection. It is necessary to have a comprehensive understanding of target disease associations, pathway target drug disease relationships, as well as adverse events profiling capability. In addition, it should be understood that target selection is either additive, targets on the same pathway, or synergistic, targets on functionally complementary pathways, in nature. 2. Drug candidate design selection. The typical approach is to integrate a different combination of pharmacophores, a molecular recognition feature of a substrate or ligand necessary to bind to a biological macromolecule, such as a target protein, into the Systems Pharmacology drug candidate. The design of the candidate drug is typically defined by the nature of the targets, the availability of starting frameworks, and the chemical tractability. Beyond that, the essential requirement of multi-target compounds is that each pharmacophore retains the ability to interact with its specific target. These challenging tasks involve consideration of structure-activity relationships that determine substrate-target interactions. In addition, establishing the same degree of affinity for each target and minimizing off-target adverse effects are critical factors to be addressed. Ramsey has argued that the rational design of multi-target compounds is far from being an easy task. Dealing with the crucial issues of selecting the right target combination, achieving a balanced activity towards them, and excluding activity at the undesired target or targets, while at the same time retaining drug-like properties. In order to achieve such a difficult task, a variety of approaches have been employed, and include 1. Computational ligand discovery. A variety of tools are used, including chemoinformatics, virtual screening, pharmacophore development, molecular docking, and dynamic studies. 2. Quantitative structure-activity relationships, QSAR, used in lead optimization to correlate molecular structure with pharmacological and biological activity. 3. Biological validation of leads, use of high-throughput compound assessment and identification of pharmacophores. 4. Receptor biology and binding affinities, screening compounds against specific targets. And 5. Cell-based assays, used as a preliminary screening tool. A number of the tools and technologies used in conventional drug discovery and development are also used in systems pharmacology drug discovery. However, the advent of the systems biology toolbox facilitated the construction of pathways or networks to afford the one-drug multi-target pathway or network paradigm necessary for the creation of systems pharmacology drugs. Systems pharmacology approaches. 2 different brief descriptions of a specific target selection and drug candidate design or selection examples will serve to highlight the approaches used in the implementation of systems pharmacology. In the former case, the identification of individual targets and synergistic combinations of targets is critical to the systems pharmacology discovery process. Most target proteins consist of more than one structural domain, but there is a limited repertoire of domain types. Since protein domains mediate drug-protein interactions, Moya Garcia and colleagues used this principle to guide the design of systems pharmacology drugs. In their study, they associated multi-target drugs with Cath functional families. Cath, CATH, is a database of 95 million protein domains classified into 6,119 superfamilies. They concluded that a small fraction of a specific functional family of proteins, Cath funfams, were druggable, and showed using structural analyses that the domains in these families have the potential to be the druggable entities within drug targets. In the second example, Thiel and co-workers outlined a detailed quantitative systems pharmacology QSP approach to determine systems pharmacology drug properties through a mechanistic consideration of processes underlying drug absorption, distribution, metabolism, and excretion, ADME, as well as the resulting drug action itself, as well as a detailed description of drug pharmacokinetics, PK, and, simultaneously, drug pharmacodynamics, PD. In this study, a QSP approach was applied to quantify the drug efficacy of cyclooxygenase 2, COX-2, and 5-lipoxygenase, 5-LOX, inhibitors by coupling physiologically based PK models at the whole body level with affected biological networks at the cellular scale. They presented results that revealed insights about drug-induced modulation of cellular networks in a whole body context, thereby describing complex pharmacokinetic and pharmacodynamic behavior of COX-2 and 5-LOX inhibitors in therapeutic situations. The results demonstrated the clinical benefit of using QSP to predict drug efficacy, and hence its value in helping to design optimal systems pharmacology drugs. Systems pharmacology drugs. The concept of systems pharmacology drugs was first suggested in the year 2000. In the intervening years, the academic and pharmaceutical communities have enthusiastically developed this paradigm, and now a significant number of such drugs are available on the market today. Lin and colleagues noted that from the year 2000 to 2015, the FDA approved a total of 361 new molecular entity (NME) drugs. They estimated that around 43% of those approved drugs had 2 or more targets. Although to be clear, such entities were not necessarily designed as systems pharmacology drugs. Subsequently, Ramsey and colleagues did a similar study in 2015 to 17 and noted that of the 101 NME drugs approved by the FDA, 34% were single-target drugs, 21% were multi-target drugs, and 10% were combination therapies. They concluded that these numbers unequivocally supports the attractiveness of systems pharmacology drug strategies. The systems pharmacology drugs approved by the FDA between 2015 and 2017 were predominantly anti-neoplastic agents, constituting 12 of the 21, 57%, approved NMEs. An additional 4 approved systems pharmacology drugs were therapies for CNS indications, with the remaining 5 drugs approved for anti-infective, musculoskeletal, genitourinary, and alimentary tract metabolism disorders. Systems pharmacology drugs are most relevant for disease states involving large target networks and pathways, as is the case with most cancers. Cancer cells are characterized by a transformed phenotype associated with uncontrolled proliferation and immortality. This abnormal cell activity is sustained by significant protein deregulation. For example, the protein kinase family consists of more than 500 different proteins, and they are involved in multiple cellular pathways and networks. The original one-drug, one-target model produced highly selective kinase inhibitors. However, it has become increasingly clear that more effective targeted therapies based on kinase inhibitors should target the disease at multiple target nodes in the network, which explains the significant efforts to create systems pharmacology-based kinase inhibitors in the treatment of several forms of cancer. The authors concur with Levy that there are 3 more general applications where systems pharmacology drugs can be expected to impact in the future. Firstly, as noted, complex disease conditions such as cancer and CNS disease are a clear focus of current effort and development. Another area of applicability is in drug resistance. The ability to simultaneously modulate different targets could be advantageous to individuals expressing intrinsic or induced variability in drug response due to modifications in key disease-relevant biological pathways and activation of compensatory mechanisms. Apart from the obvious applications in the field of antimicrobial chemotherapy, it is less probable to develop resistance linked to single-point mutations against multi-target than single-target agents. This strategy could also be pertinent to treat non-infectious conditions characterized by high incidence of the drug resistance phenomena, for example, epilepsy. One final application for consideration is in prospective drug repositioning. We have previously highlighted the resurgence of drug repurposing and repositioning, DRPX. However, most such cases have occurred by serendipity or exploitation of the original mechanism of action, using a retrospective DRPX approach. Levy has argued that in contrast, prospective DRPX should explore drug repositioning possibilities much earlier in the drug discovery process. This would entail the design of multi-purpose drugs to treat different indications, such as comorbidity disorders, for example diabetes and cardiac disease. Systems pharmacology conclusions. The reality of the one-drug multi-target pathway on network systems pharmacology drug appears to afford considerable advantages over the one-drug, one-target or combination drug multi-target approaches. However, the tremendous therapeutic potential of multi-target drugs, their rational discovery, and their development still represent a formidable challenge. In addition, the necessity of balancing the beneficial polypharmacology versus the harmful promiscuity of such drugs still needs to be evaluated as more of these drugs are brought to market. Nonetheless, building on the accumulated evidence, the concept of systems pharmacology drugs has made rapid and spectacular progress from being an emerging paradigm when first enunciated at the beginning of 2000 to one of the hottest topics in drug discovery and development over the last couple of years. Does the merging of systems biology, precision medicine drugs, and now systems pharmacology drugs provide the harbinger of more safe and efficacious drugs? Initial indications appear to suggest that there is indeed a new dawn breaking for patients and the products they rely on to keep them healthy and alive. This article was written by Dr. Kirkwood A. Pritchard Jr., Dr. Dustin P. Martin, and Dr. Stephen Naylor. Dr. Kirkwood A. Pritchard Jr. is a co-founder, CSO, and board director of ReNeurogen LLC, a virtual pharmaceutical company developing systems pharmacology therapies for the treatment of neurological diseases. He is also a co-founder and board director of HDL-DX, a diagnostics company developing an assay to determine functionality of HDL. Dr. Dustin P. Martin is a research scientist at the Medical College of Wisconsin. He has served as adjunct professor of biology at both St. Mary's University and Winona State University. His current research interests include systems pharmacology predicated on peptide-based anti-inflammatory peptides in a number of neutrophil-mediated diseases, such as stroke, traumatic brain injury, sickle cell disease, and bronchopulmonary dysplasia. Dr. Stephen Naylor is a co-founder, CEO, and board chairman of ReNeurogen LLC, a virtual pharmaceutical company developing systems pharmacology therapies for the treatment of neurological diseases. He is also a co-founder, COO, and board chairman of iMetabolic Biopharma Corporation. In addition, he is the founder, chairman, and CEO of myHealthInc, a systems network biology level diagnostic company in the health and wellness and precision medicine sector. If you've enjoyed this episode, then you can subscribe to Drug Discovery World free of charge by visiting our website at ddw-online.com, where you can also view all of our articles including the references and images, and download original PDFs. All the links are in the show notes. If you're enjoying the podcast, then do leave us a review and subscribe, and you can also follow us on social media on LinkedIn and Twitter. Thanks for listening, and we'll hope to see you in our next episode.